Okay, so you were trying to get the pictures of the parathyroid and okay. And yeah, that's a good one. Also, what the APR is good for is to get your thyroid, not thyroid, your um, pituitary gland complete, because it has some terms on there that are not found in your textbook. So the pituitary gland is very good for a histology part. Okay, and we have a couple more people that are coming. Alyssa, if I could find you. Oh, there she is. Oh, no, she disappeared. And it does help me a little bit if you put whenever you sign in, if you put your last name in there as well. That way it's a little easier for me to find you on my roster. And again, I'm just checking just so that I can have a record of who was here. You're not gonna get like penalized or anything for not being here because you can watch that on your own. So, but it's just to keep track of things. All right, so let's go ahead and start talking about chapter 18. Hopefully you have gone through your endocrine system recordings. So normally, like if we were in class, normal class, I will do chapter 17 with you pretty much in one day, or most of that chapter 17 would be done on the first day. And then we would finish 17 and start 18 on the second day of our 2 meetings that we would normally have in class, right? And so here you had the endocrine system. You had it broken down into 3 sections. Hopefully you're caught up with that. Yeah, we're gonna start talking about blood here. So I'm gonna share my screen here with you. And that's this. Let me see if I can get rid of this just so we have fewer things on there and a bigger view of this. So this is just the first initial image here. It doesn't really tell us too much about anything, but it's a picture of blood during a transfusion. Okay, so blood is a fluid connective tissue, and its major function, like when people think about blood, most of the time they think about, oh, blood flows through our blood vessels and carries things throughout through our body, which is true. That is the most known function of the blood to general people. However, it's going to have a bunch of other functions as well. Okay, so blood does transport various materials. We know that blood transports hormones. We just talked about hormones, and we said that hormones travel through blood. Okay, so that's one of the things that are being transported through blood. Blood is going to transfer nutrients and wastes and respiratory gases like oxygen and carbon dioxide. And so it transports various things. And some are going to be transported by the means of cells like your erythrocytes, and some things are going to be transported as dissolved in the blood plasma, okay? So since I mentioned blood plasma, the blood, if you look at whole blood, blood is composed of 2 main parts. And actually we'll divide whole blood, like when you look at the test tube, it's going to be divided into 3 parts. But in general, the blood is divided into 2 main components, and these are your formed elements. And formed elements are going to include the solid particles. These are the cells like your white blood cells, your platelets, and your erythrocytes. Okay, so these are cells or fragments of cells, which this is what platelets would be. Erythrocytes are kind of in there, but these are the solid particles. Okay, cells or remnants or fragments of cells are formed elements. And then the second part of the whole blood is going to be the fluid part, which is called the plasma. Okay, so this is the fluid part of the blood, and this is going to contain the dissolved particles. Okay, so formed elements and plasma, 2 components of blood. Okay. The functions of the blood, and I know this picture there's not really a whole lot of pictures for chapter 18, okay? So this is just going to show us characteristics of blood, which I will get to in a minute. But before we get to the characteristics of blood, and I guess I don't really need to be sharing that picture with you yet. So the functions of the function of the blood can be divided into three main things. First function would be the transport. Second function would be the regulation. And then the third function would be protection. So let's go ahead and look at each part of this function, kind of an umbrella of blood and what the blood does. So as far as transport goes, I've already mentioned that blood is going to transport dissolved particles and formed elements and various components, okay? So there's not really much to add to that. I think that's pretty self-explanatory as far as transport goes. The second part of the function section here is the regulation. And you can actually look at regulation and you can divide the regulation into three subparts here because blood can regulate your pH, it can regulate your body temperature, and then it can regulate fluid balance in your body. It's a very important thing in regulation of fluid balance. So for the pH of blood, and that would be something that was in that table. Oh, somebody else is coming. The pH of blood is between 7.35 and 7.45. So it's a very narrow range for pH, and it needs to remain in this range in order for the proteins to function correctly and for them to not get denatured. So if the blood gets too acidic or too basic or alkaline, it would cause proteins to get denatured. And this is bad because when your proteins get denatured, they are no longer able to function like they need to. Okay, so that's the pH. So the way that the blood regulates pH and keeps it in this narrow range is by containing buffers. So in plasma, we can find buffers, and buffers are chemicals that are able to absorb excess acid or base, so that when following a meal, you're absorbing nutrients. You're going to absorb some things that are acids and bases, you know, depending on what you're eating. As all of this is entering your blood, it doesn't cause a sharp spike initially because you're going to have these buffers that are going to be able to bind this. So think about buffers as sponges for extra acids and bases. And we'll discuss this more in chapter 25. So that's the pH. Blood can also regulate body temperature. The temperature of the blood is actually about 1 degree Celsius higher than your measured body temperature. And so as the blood travels through your body, it warms up your body. We will see that we can actually regulate the blood flow to different parts of your body, whether we are trying to conserve heat or release heat. So following an exercise, you're going to see the blood being redirected to the capillaries that are underneath your skin. And that would be because you know, blood carries the heat with it. And so if it's allowed to go to the skin, you can release that heat through the skin. So that's regulation of body temperature. And the last thing that blood regulates is fluid balance. And the way that it regulates fluid balance is by the concentration of different things that are in the plasma. When you think about this, and again, a little more of this will make sense when we actually talk about capillary exchange, and that's going to be in chapter 20. For now, just know that things move in and out of blood depending on the concentrations of particles that are in the blood versus what the concentration is outside of the blood, like in the tissues, okay, in the interstitial fluid. So you're always going to have kind of a relationship between what's happening in the blood and what's happening in the interstitial fluid. And depending on the concentrations of things on the two sides, water is going to either move out of the blood into the tissues or the other direction. So if the blood is more concentrated than interstitial fluid, then the water would be moving into the blood to trying to balance that out, and vice versa. So if you have the interstitial fluid more concentrated, then water moves by osmosis into that higher concentration of solutes. Okay, so we say that blood exerts a colloid osmotic pressure, or well, the substances that are in the fluid exert colloid osmotic pressure. We will mention it again in chapter 20, okay? So people don't like the colloid osmotic pressure at all. I don't know why. It's really not that bad, but I think once we put it into perspective with the capillary exchange, it's a little easier to understand. For now, just know that things move in and out based on concentration of solutes. So that's a regulation. So again, blood regulates temperature, pH, and fluid balance. And then the third thing of the blood's function is protection. And protection, this is kind of a two-way thing here. Blood contains leukocytes. Leukocytes are your white blood cells, and these are your immune cells. And so that's part of the immunity right there. The other part of immunity is the antibodies. So blood contains antibodies. Antibodies are proteins that are designed to fight a specific type of pathogen. We will actually introduce antibodies a little bit in blood typing. So that lecture is already posted, and it follows, you know, whenever I finish this, you would look the blood typing first, and then hemostasis, which is the last part. Okay. And so antibodies are proteins that are designed specifically against a specific type of foreign substance. Okay. So in the blood typing, you know, you'll see that you have antibodies against the blood that you don't have. Okay. A blood that's foreign to you. But we have antibodies against specific bacteria, specific viruses as well. Whatever we have been exposed to, we form antibodies as part of our immune response against that. Okay, so that's protection. Okay, let's talk a little bit about the characteristics, and this is where the table kind of comes handy here. So let's look at this table here. So the color of the blood is red. Blood is not blue. Okay, it is the oxygen-rich blood is going to be bright red. Oxygen-poor blood is going to be dark red. So if somebody is having bleeding and they're trying to figure out whether it was artery or a vein damaged, well, I mean, you can tell because artery is going to be shooting. There's higher pressure in arteries, okay? And the color of the blood would be also different. The venous blood is going to be dark red. But blood is not blue. So when you're looking at your blood vessels right here, they look like they're blue. If you have read your chapter already, they explain the fact that your skin kind of acts like a filter, and your eyes can only see the light that hits your eyes, right? So your skin absorbs the shorter wavelengths of light, like those are the reds. The blues and purples are the longer wavelengths, and so they are actually able to pass through the skin, and so that's what you see, but it's not actually blue. If you were to cut it, it would not be blue. Okay, so that's our color. For volume, our blood volume is about 5 liters, and this tends to be a little higher in males than females, and on average, and that's because an average male is larger than an average female, and so men tend to have a little higher blood volume. And also men also tend to have a little higher blood cell percentage, like higher hematocrit. And that has to do with testosterone. It's one thing that affects your percentage of erythrocytes in your whole blood. Viscosity would be another characteristics that we should talk about. Viscosity is thickness of the blood. If blood is water and some other things like cells and solutes, that naturally means that blood has to be thicker than just pure water. I heard a sound, and I don't know if that's a chat or what. Nope. Maybe somebody left. I don't know. But anyway, so viscosity is thickness. Blood is anywhere between 4 to 5 times thicker than water. And this changes, it changes constantly. Water is not going to have a constant composition. It has ever changing composition, because anytime you take a drink of water, for example, that water goes into your digestive system and then it's absorbed into your blood and it dilutes your blood. And so it would be a little less thick, right? And then your body loses water all the time. So, you know, it's kind of a relationship or kind of a balance between, you know, water loss, water gain, but solutes as well, you know, or things that are in your blood, they're being carried in your blood. These are going to change constantly depending on, you know, have you just eaten? If you just ate, your body is going to be absorbing all those nutrients and things are going to be rising in the blood. And so that's viscosity. And that kind of goes hand in hand with the plasma concentration as well here. And the plasma concentration simply refers to how many things you have dissolved in your blood. And again, it constantly fluctuates, okay? Blood temperature is, I've already mentioned that blood is a little warmer than your measured body temperature, okay? So it warms up your body as it travels through. pH, I've already told you the range, 7.35 to 7.45 range that we need to have. There she is. Okay. So let's look at the components of blood, and that's this next image right here. So we'll stay on this image for quite a while. So this image shows you centrifuge. Centrifuge is an instrument that spins really fast, and we use centrifuge to separate contents of a test tube or a container based on their weight and size. And so here the erythrocytes are going to be your heaviest things, so they're going to be at the bottom of the test tube as you spin this. Then in the middle part of the whole blood right here, that kind of the white band or kind of grayish, I guess, that's your buffy coat. And then the rest of the whole blood right here, that's your plasma. And so I do want you to know the distribution of the components of the whole blood. Okay, so I want you to know that blood is mostly plasma. Whole blood is mostly plasma. And then only about 45% are the formed elements. Okay, these two right here, the red and the grayish little thin band, these are your formed elements. Okay, so those are your formed elements. Oh, why didn't it finish writing? Okay, formed elements. So majority of your blood, again, is going to be plasma. Now, if I ask you what is the most abundant formed element, you will not say plasma, because plasma is not a formed element, correct? Okay, so pay attention to what it's asking. Like a question might say, what is the most abundant component of the blood? And then you would say plasma. What is the most abundant formed element? You would say erythrocytes, okay? I mean, you can see right away, formed elements are the red part and the gray part, right? What's the bigger one out of those two? Obviously, it's the red one, right? And so erythrocytes are your most abundant formed element. Okay, let's see what we have next. Okay, so let's talk about these parts of the whole plasma a little closer. Now, when we look at the individual parts, I do not need you to tell me the percentages, but I would like you to know what is the most abundant part of that each section, okay? So plasma, okay, looking at the top box right here. I want you to know that plasma is 55%-ish of whole blood, okay? That number I do want you to know. I want you to know that plasma is mostly water, okay? You don't have to tell me 92%. I want you to know that it's mostly water. And then we have proteins that are in the plasma, and then we also have solutes, other solutes that are found in plasma. Okay, from the proteins, we have the most abundant protein in the plasma is albumin. You don't have to tell me 58%, but know that that's the most common protein in our plasma. It's the albumin. Albumins are transport proteins. These are going to be the ones that most likely will serve as the protein carriers for lipid-soluble hormones. Okay, does that ring a bell? Some hormones are lipid-soluble and they need a carrier protein. Well, albumin is a carrier protein. It's a great one for this. Okay, so that's albumins. The next group of proteins are globulins, and globulins are also transport proteins. There's a special group of globulins which are known as immunoglobulins or your antibodies. That's going to be related to antibodies there. But mostly, I mean, they're transport proteins as well. Then we have fibrinogen, and fibrinogen is not a huge percentage, but it's worth to mention here because fibrinogen is going to be important during hemostasis, and hemostasis is blood clotting. Fibrinogen is a soluble protein in this state when it ends with a gen, but during a blood clotting pathway, it eventually gets activated and converted into a fibrin, and fibrin is going to be non-soluble. It's going to create like a framework for a blood clot. And then we have some other regulatory proteins in there that we're not going to name, but these are your main ones. These three are your main proteins in the plasma. Some other solutes that you might find here are electrolytes and nutrients, gases, and waste products. Okay, so the stuff that generally gets passed along through our body by means of blood. You would also find hormones there and such. So that's our plasma. Okay, let's look at the buffy coat, which is the part of the blood that contains platelets, and it also contains your leukocytes. So you don't need to, again, know the numbers. Know that buffy coat is about 1% or less of whole blood. Yeah, that's that. For the leukocytes, I want you to know that the most abundant leukocyte is which one? If you look at the numbers here, you can see that these guys right here are the most abundant. So your neutrophils, but you don't have to tell me that it's it's percentage. It's generally more than half of your leukocytes in the blood are neutrophils. For that reason, generally these guys are first at the site of infection, generally, because they're the most abundant in your blood. However, notice how few, like how small that buffy coat is. Leukocytes are larger than erythrocytes. So if you look at an individual leukocyte and then the erythrocytes is little compared to the leukocyte. Yet buffy coat is so small. And this is because leukocytes actually use the blood to just travel, like go somewhere. And there are normal values of leukocytes in your blood that normally should be there. But majority of your leukocytes are going to be in your tissues somewhere else in your body. So your body has way more leukocytes than what you see here in the blood. They're just not, blood is not the primary place where you find leukocytes, okay? So you're gonna find them in your lymphatic tissues and organs, in tissues is where most of them are found. But some of them need to be in the blood as well. So for your neutrophils, there's a table. Let me see, where is it? Since we're talking about white blood cells, this one right here, okay? And so what I would like you to know for these leukocytes is what they do. You don't need to worry about characteristics. I'm not going to make you identify them. Your Learn Smart might have you identify them, which is when you reach the book and look it up, okay? But on the exam, I'm not gonna make you identify these based on characteristics. So I want you to know what their functions are. Okay, so for neutrophils, just to kind of summarize this, for neutrophils are engulfing cells. They like to phagocyte, or they're phagocytes. Okay, so they engulf mostly bacteria. They prefer bacteria. They can engulf other things too, but they prefer bacteria. And then they destroy them. They kill them. Eosinophils are the next on the list, and these are going to play a little, like a small role in an allergic reaction, but these are going to be really good at targeting multicellular pathogens, such as like worms and stuff. They can also phagocytize antigen and antibody complexes and allergens, yeah. So that's that. Basophils. Basophils are going to be important in allergic reaction. They are a little more significant in allergic reaction. They release histamine and heparin. Histamine is going to be vasodilator. So when you have an allergic reaction to something, you know how like your eyes are all like runny and your nose is runny. This is all that vasodilation of all those vessels is causing all this fluid to leak out of you. Okay. Because of that histamine that's released by basophils. Heparin is going to prevent blood clotting, and this is going to be important in yep, I'm totally blanking out on this term. Inflammation response, oh my gosh. I'm like, it starts with an I. But yeah, inflammation response is regulated by basophils, and it's aided by basophils. And look, it's like in that table, and here I am trying to think about it. Could have just read it. But anyway, so those are basophils, and these three are granulocytes. So I would like you to be able to classify granulocytes and agranulocytes. It's easy way to remember which one is which. The granulocytes are all the phils, okay? So neutrophil, eosinophil, basophil, all the ones that end with phils are granulocytes. And then the other ones are agranulocytes. So we have our monocytes and lymphocytes. Lymphocytes, actually, we're going to split in our immune system chapter into B lymphocytes and T lymphocytes. And T lymphocytes are going to be split into helper and cytotoxic there. And then we also have, yeah, natural killer cells are also a type of lymphocytes. But anyway, so these, because we can separate them, each group of lymphocytes is going to do something quite different from each other. So it's almost hard to summarize that. But yeah, I guess saying they coordinate immune response or immune cell activity and produce antibodies and all that. That's a good kind of summary of that. But again, eventually later on, you'll be able to tell me what each specific type of a lymphocyte does. So for now, this is okay. So by the time we get to chapter 21, this is going to be a review for you. You're already gonna know what these leukocytes do. And then monocytes are cells that become macrophages. These are going to be cells that are large cells that engulf anything that needs to be destroyed. So this can be bacteria, viruses, it can be sick cells, remnants of cells, debris that's not supposed to be there. Macrophages do that. So that's what monocytes become, these macrophages. So going back to this image right here, okay, since we're already on leukocytes, I kind of wanted to, talk about them there. And the last thing, I guess, out of this is going to be the erythrocytes. And erythrocytes are the most abundant formed element. Write it down and highlight it and say it to yourself over and over and over. The most abundant formed element of blood are erythrocytes, okay? So over and over, keep saying that. Let me see. Let's look at erythrocytes. And you know what? Let's actually, since we are going to talk about erythrocytes, let me go here to the hemopoiesis, and that is formation of these cells, and then that will lead us into characteristics of erythrocytes. Before we get there, this is an image of a blood smear. So some of you were asking where you can find the histology file. I have been having so many issues with trying to load things onto Blackboard. It's saying that I'm loading too much stuff, uploading too much stuff in there, and I've exceeded the size or whatever. So I have posted a link to a Google Drive. This link will only work for you if you're signed into your browser with your TCC credentials. So make sure that you're signing in with your TCC credentials, and it should open the PowerPoint, the blank PowerPoints, my old PowerPoints with like words, kind of like notes, and then the histology file is there as well. Okay, it is a large file, so if you're trying to download it, it might take you a long time, but be patient, it should download. And that's one reason, I guess, why I wasn't able to load it on Blackboard, but Blackboard wouldn't even let me upload all the PowerPoints into a folder today. It said it was too big. So Google Drive took it, and I hope that you have access, you can access that just fine. Once you try it, let me know if it doesn't work, okay? Okay, I see a chat notification. Okay, perfect. Okay, so I guess it works. That's good to know. That is good to know. All right, so let's, so anyway, you can use for your histology, when you're drawing the histology stuff into your lab exercise that you are turning in, you can use a picture from the textbook, that's fine. You can also Google a picture at a specific magnification, or you can use the APR, and the magnification, the APR doesn't like obviously tell you the magnification, but if you are using that, and if you click on image info, it will tell you what the magnification is. You can draw that in and just say, okay, this is not 40, this is whatever, 100, and just draw that. So anyway, you have options. As long as, as long as it's blood, if the slide is asking for blood, as long as it's blood, it should look pretty much the same no matter where you're looking, okay? So that's that. So this would be a typical blood smear, what a typical blood smear would look like. We would not be doing this in actual lab. Like we wouldn't be doing your blood. You would just look at an existing one. And you can see the size comparison of your leukocytes. See this, these are the leukocytes. They're different in color and they're also different in size compared to erythrocytes, which are these. See, all these are erythrocytes. And when you look at the erythrocytes, they look kind of like they're see-through in the middle. They are not see-through in the middle. They are just kind of squished in the middle, okay? So that makes it look like they're see-through, but they're like an incomplete donut almost, okay? It's a biconcave disc is the actual proper term for it. So it's like if you had kind of like a, like something almost round and you squished it in the middle. That's what it looks like, the erythrocytes. Okay, let's go. That just summarizes things that are in there. You don't need to worry about this, maybe just for reference, but I'm not going to test you on these values here. Okay, I want this table right here, okay? So this table, this is hemopoiesis, and hemopoiesis is formation of formed elements. Okay, remember that definition. Hemopoiesis is formation of formed elements. Do not get it confused with hemostasis. Hemostasis is formation of blood clots or blood clotting. Okay, so hemopoiesis is formation of formed elements. There are several things that I want you to know from here, but there are several things that I'm not going to ask you, like these colony stimulating factors. I'm not going to, and the intermediate steps here for the leukopoiesis, I'm not going to make you do. Erythropoiesis and thrombopoiesis, there's a little more detail that I will request you to know. Okay, first thing that is important. Let me get a different color, maybe. Okay. Well, first of all, know that you know. I mean, make sure that you know this this term, the hemopoiesis. That's what it is. I want you to know hemocytoblast. I want you to know that that's the cell that is your blood stem cell. Okay. So this is the cell that gives rise to all the formed elements in your blood. Hemocytoblast. Okay. Hemocytoblast can be stimulated to either go through a myeloid line or lymphoid line, okay? And we're not getting into details as far as what stimulates it, but know that there is a signal, there is a chemical signal that tells this hemocytoblast whether to go to the left or to the right, okay? Whether to go into the myeloid process or the lymphoid process, okay? In this case, the word line means like a process. Okay, So I'm going to start with the lymphoid line because it's easier, okay? It's simpler here. The lymphoid line only leads through one process, and that is the process of leukopoiesis. And so the lymphoid line, through the process of leukopoiesis, gives you B lymphocyte, T lymphocyte, and natural killer cell. So if I asked you, what are the three types of lymphocytes? You would say B lymphocytes, T lymphocytes, and natural killer cells, okay? Natural killer cells don't sound like lymphocytes, but they are, okay? And they're all these three are from the leukopoiesis from lymphoid line. You can see that leukopoiesis also comes from myeloid line, right? So a cell, the blood stem cell, can go to the myeloid line and then enter the process of leukopoiesis as well. But from the myeloid line, leukopoiesis is going to give you the FILs and a monocyte, or monocytes, not a monocyte, many monocytes, okay? So leukopoiesis from myeloid line gives you the granulocytes and monocytes. So all the FILs and monocytes. So I want you to know leukopoiesis from lymphoid line gives you those 3 lymphocytes. Leukopoiesis from the myeloid line gives you those 4 cells. Okay, see, simplified. I'm not going to ask you about those intermediate cells in there for leukopoiesis. Okay, I think, are we good? Yes, maybe? Thumbs up? Yes, no, there's like a green yes that you can say. Okay, fine. Don't talk to me. So anyway, so that's leukopoiesis. All right, let's stick with the myeloid line for some longer. The myeloid line can also lead to the process of thrombopoiesis. Okay, and the process of thrombopoiesis is formation. Yay, I got a thumbs up. I got a, so anyway, so myeloid line can go into the process of thrombopoiesis, and thrombopoiesis is formation of thrombocytes, which is a fancy word for platelets. And here, I would like you to know thrombopoietin. Thrombopoietin is a stimulating factor. It's really a hormone that is going to affect the formation or development of this megakaryocyte. So that word I do want you to know as well. See, megakaryocyte, and it looks like this. It's a very big cell. That's why it has mega in the word, right? And megakaryocyte has these things that kind of stick out of it or hang out of it. And these are called proplatelets. So you have them zoomed in right here, like enlarged. And these proplatelets, here's what happens with these guys. Okay. When you look at this picture, and this is somewhere nearby of the hematopoiesis, this shows you the visually the process of thrombopoiesis. So you would have a megakaryocyte in here, and this is in the red bone marrow, by the way. So megakaryocytes don't actually get into your blood, but they're going to be on the outside of a capillary. Okay, this is a capillary. It's just a single row of cells, and so megakaryocytes will sit on the outside of the capillary. Okay, so this right here is the outside of the capillary. And those proplatelets will eventually stick through the gaps between the cells. So even if there are not really necessarily gaps between the cells, the proplatelets will kind of push themselves between the 2 cells, okay? And they will stick inside like this, okay? And as the blood flows through that area and these proplatelets are sticking to the inside of the blood vessel, they literally break off those proplatelets. They break off pieces of the proplatelets. So platelets are really just fragments, small tiny fragments of the megakaryocyte. It's like a little piece of cell membrane and a cytoplasm in there. Okay, there's other stuff in there too, and that has to do with the function of the platelets, but that's kind of how they get chopped off. And eventually the megakaryocyte becomes smaller and smaller and smaller and smaller because it gets chopped up into little platelet pieces. Okay, so that's how platelet formation works. So know the thrombopoietin and know the term megakaryocyte, because that's what gives rise to the platelets. And kind of just be generally aware of how, you know, those platelets get chopped off. The last thing, erythropoiesis. Erythropoiesis is formation of erythrocytes, and these are the red blood cells. Erythropoietin is an important player in formation of the erythrocytes. Erythropoietin comes from kidneys, let me see. Erythropoietin comes from kidneys. It is stimulated by low oxygen conditions, as well as testosterone. And this is the testosterone presence is one reason why males tend to have a couple percent higher hematocrit or percentage of erythrocytes in their blood than females. Environmental factors can also affect erythropoietin release, which kind of has to go, like when you think about low oxygen condition, if you're healthy, the low oxygen conditions you would experience if you're like at a higher altitude. So just last week, we were in Colorado Springs, so that's about 6,200 feet above sea level, something like that. So that was a little, that took a little while to get adjusted. And then we ended up in New Mexico, and the place we were staying at was 10,000 feet above sea level. So that one you definitely felt. And so I guess maybe our percentages of erythrocytes should be a little higher, probably, because we're exposed to these low oxygen conditions in the higher altitude. So anyway, so that's stimulus for the erythropoietin release. What I want you to pay attention to as you're looking at this pathway of the erythropoiesis, notice that the cell begins at a certain size, and then by the time you get all the way to the bottom, it's much smaller, and it already has that typical shape of an erythrocyte. It is also getting more and more red, and the redness comes from the protein known as hemoglobin. Okay. Hemoglobin is a protein that is made out of 4 globin chains. And since I'm talking about it, let me show it to you. Okay. So we have 4 globin chains, 2 alphas and 2 betas. And each of these globin chains has this heme group. And inside of the heme group is a porphyrin ring. Okay. So this is because it's kind of like this, we call it a ring. And inside of this this heme group, you have an iron atom. So Fe is iron, and it's an atom in there in the center of that heme group. And the iron is what binds oxygen. Okay, so hemoglobin carries oxygen. Hemoglobin can also carry carbon dioxide, and it can also carry hydrogen ion. So hemoglobin is not only designed to carry oxygen, but oxygen is kind of the main guy here that gets carried by hemoglobin. And it is the only thing, oxygen is the only thing that binds to this iron here. The other things could bind to hemoglobin, but they don't bind to that iron, okay? The iron atom is what gives the erythrocytes that red color, okay? So it's that iron where the color comes from, okay? So that's hemoglobin. So as the cell is as the erythrocyte is formed, the cell becomes smaller and smaller. You can see that eventually it ejects its nucleus. It gets rid of the nucleus. What is not shown here is that it also gets rid of most of its organelles, and the very last organelles to go are going to be the ribosomes. The reason for this is, if you remember from AMP1, what ribosomes do, ribosomes are going to produce proteins. Hemoglobin is a protein, so we're going to hang on to those ribosomes for the longest time, and then towards the end we can toss those as well. So what you actually end up with at the end, when you have a final erythrocyte, is it's a cell membrane, okay, the plasma membrane, and then cytoplasm and hemoglobin. That's it. That's your erythrocyte. So technically the term erythrocyte is not really proper because cyte means cell, and this thing no longer has nucleus or organelles. So we really consider it or call it cell incorrectly. So that's erythrocytes. So because erythrocytes do not have nucleus, they are going to have a defined lifespan. They don't last forever. They get worn out and they cannot reproduce because they don't have nucleus, and there's nothing that will regulate the reproduction. The erythrocytes are, or you know what I forgot to mention, where hemopoiesis happens. Hemopoiesis happens in the red bone marrow in adults. And then during like fetal development or embryonic development, we see the first blood cells come from the yolk sac during embryonic development. And then as organs begin to develop, we see a hemopoiesis happening in a spleen and liver. And then once the fetus has bones, eventually it will be bones, the red bone marrow of certain bones. So that's that. The erythrocytes have a lifespan of about 120 days or so, and they are going to be degraded by macrophages, mostly in spleen and in liver. When they're broken down, the cell membrane is just broken down into components, and then we can recycle those components to create new cell membranes. The hemoglobin breakdown can be a bit tricky. The hemoglobin breakdown, we have to break it into the globins, the globin chains. They're proteins, so they're broken down into amino acids. The heme is broken down into bilirubin and biliverdin. And bilirubin, accumulation of that is actually what gives you like the yellow color during jaundice, and that's like a sign that your liver wouldn't be functioning correctly. And then the iron, we actually recycle most of the iron from the erythrocytes, and so you do lose some in urine and feces, but most of it is recycled. And for this reason, iron, even though it's important for hemoglobin, it is not considered one of our major minerals that we need to consume daily. But also, on the other side, it's one of the most common deficiencies as far as electro or minerals go. So, but in a well-balanced diet, generally we have enough iron, and our body recycles most of it. So you don't normally need to consume a lot of it. Because a lot of iron is bad too. So that's erythrocytes and hemopoiesis. I think the next part will go into, this shows you the size of the erythrocyte. There are some spectrum proteins those are the ones that are responsible for holding the shape of the erythrocyte. And then erythrocytes can arrange themselves into this Rollo kind of arrangement where they create these rows here, and we have a name for that. So but basically the size of a capillary in your body is about the size of an erythrocyte, so capillaries are very, very small. Okay, that's the hemoglobin, and we kind of talked about the erythropoietin already, but this figure shows you the pathway of how erythropoietin would be stimulated and kind of what happens. I've already mentioned that it comes from the kidneys. So anyway, so that's that. This shows you sickle cell erythrocytes, and this is a condition. It's a genetic condition where your hemoglobin is not correctly formed, and then that is going to affect the shape of your erythrocytes that become sickle celled like this, and they are less efficient in carrying oxygen, and they can also get stuck in your capillaries. So people have these things known as crises, and it's not very pleasant if you have a full-blown sickle cell anemia. We'll actually revisit sickle cell a little more in genetics section, because it's an example of codominance if you are heterozygous for this trait. If like one parent gives you sickle cell and the other parent gives you regular hemoglobin. Okay, this is kind of a summary of everything that we have talked about. And then this shows you transfusion, and then it goes into blood typing. Yep, so this is where I'm going to stop for now. You have 2 more videos to watch. You have the blood typing and you have the hemostasis to watch. So they're already uploaded for you on Blackboard. What else? I did post of it like on the assignment that you're supposed to turn in, the lab manual exercise. There is a little video that talks to you about how to do that. So make sure that you watch it. It's not super long, okay? I'm giving you hints how to do it, so do it right. Yeah, and then do the blood typing. Make sure that you're turning everything in, you know, as on time. And I'll look and see if anybody has turned in the actual lab exercise that I have to manually grade. One other thing I wanted to remind you is that the visible body assignment, I accidentally made it invisible bodies worth 6 points. It's supposed to be 8. Okay, so I'll just, you know, recalculate your grades out of 8. What else? Oh, those I have to manually import. So I have to transcribe them into Blackboard. They don't automatically go into gradebook. So if you don't see them in gradebook, don't freak out. If they're invisible body, you have the grade. I just haven't put it in yet. Okay. Okay, I think that's all I have for now. I'm gonna actually stop the sharing and I'll stop the recording and I'll see if you guys have any questions.